Hello everyone. Today I'll start with explaining the anatomy of the basic ganglia, and I'll try my best to make it as easy as possible. Now, the main goal here is not just to memorize certain structures, but it's also to understand what they actually do and what happens when they are damaged. And by the end of this lecture, we need to understand the extrapyramidal system, the deep nuclei, and most importantly, how to differentiate between the clinical cerebellar lesions. So we'll start first with comparing the pyramidal and the extrapyramidal systems, and then we're going to move to the basal ganglia, the anatomy, the pathway, the clinical disorder, and then we will look at the cerebellum and its clinical manifestations. So before we start with the basal ganglia, we're going to start again with the pyramidal and the extrapyramidal system. So first, for the pyramidal system, it's mainly responsible for the voluntary and the skillful movement. And it uses two order of neurons. It uses basically the upper motor neuron and the lower motor neuron. And it originates where? In the primary motor cortex in area 4. Now, for the crossing, 80% of it will cross through the lateral corticospinal tract, which will control what mainly? It will control the peripheral muscles, okay? And about 20% of it will remain uncrossed through the ventral corticospinal tract to control the axial muscles. So the majority controls the peripheral and 20% controls the axial. Now, another important point is the maturation. So the pyramidal system does not fully function until what? Until the first 2 years of life, because this tract needs time actually for myelination. So this is why, or when does it become fully mature? Until the first 2 years of life. Why? Because of the myelination. So if the pyramidal system is damaged, what do we expect to see? We mainly expect to see weakness and paralysis and spasticity. Now let's compare it to the extrapyramidal system. The extrapyramidal system is more involved mainly in the gross movement, such as the muscle tone, the posture, and the balance. Now, unlike the pyramidal system, it's basically multineuronal relay system, and we find it in the premotor cortex, area 6, and here it's area 4. Okay, so there is no direct connection to the spinal cord. Instead, it's basically multiple relay neurons. So it actually, some of it will cross and others won't cross, and some will be excitatory and some will be inhibitory. It functions before birth. This one we said until the first 2 years of life, the maturation. Here, it functions even before birth. Now, clinically, a lesion here is going to be definitely different from the pyramidal lesion. So instead of causing paralysis, it will cause for me what? Movement disorder, dyskinesia, and tremors and rigidity. So the easiest way to differentiate between them is saying that the pyramidal is mainly for the voluntary and skilled movement, whereas the extrapyramidal is mainly for the gross movement, for the tone, for the posture and balance. Now, let's talk about the extrapyramidal nuclei and the basal ganglia basics. Now, the most important and the largest nucleus at the subcortical level, okay, is going to be the basal ganglia. So keep that structure in mind because we'll be focusing on it based on this lecture. So it's in the subcortical area. We can organize the extrapyramidal nuclei into 3 levels. So first we have at the cortical level, we have the premotor cortex, which is for area 6, and the prefrontal cortex and the cingulate gyrus. Okay, so yes, so this is for the cortical area. And then at the subcortical level, we're going to have the basal ganglia, the thalamus, and the subthalamus. And then at the brainstem level, we're going to have the substantia nigra, the red nucleus, the pontine nuclei, and the reticular formation, the vestibular nuclei, and the olivary nucleus. Now, the important idea here is that the extrapyramidal system is not actually one structure. It's basically a network of structures, including the cortical, subcortical, and the brainstem, in order to work together and regulate the movement. Okay? So if we take a look here, we can see here the caudate nucleus, and we're going to talk about it more in depth. The putamen, the globus pallidus, the thalamus, the bradynucleus, the substantia nigra, the reticular formation, and the olive. And this is the cerebellum. Here in this picture, we can see what? We can see the globus pallidus. We can see the internal part and the external part. We can see here the putamen, and we can see the caudate nucleus, the head, body, and tail. And yeah, we have two actually definitions, the corpus striatum and the lentiform nucleus. And again, we're going to see that later on. So we're taking it step by step. Now, let's move on and talk about what exactly are the basal nuclei or the basal ganglia. They're basically what? They're islands of gray matter. Where can we find them? We find them inside the white matter of either the forebrain or the midbrain. And their main role is to control the movement. Now, in order to remember the components, we need to have this mnemonic, which is called the BBG, Amy, or baby girl Amy, called the cops on Santa Claus. Mm. So this represents what? The basal ganglia, okay? It represents the basal ganglia, the amygdaloid body, the corpus striatum, and the claustrum. So again, the basal ganglia, the amygdaloid, the corpus striatum, and the claustrum. And one important note is what? To note that the amygdaloid body is basically, anatomically, we consider it part of the basal ganglia, but function-wise, it's associated with the limbic system, and we're going to have a video about the limbic system later on. And it's very important to know that what is the claustrum, the one that we're talking about? The claustrum is basically a sheet of gray matter which is found inside the insula. So here, so it's a sheet of gray matter, okay, that is located in deep to the insula. So this is the claustrum. And in order to not get confused, anatomically, these structures will be grouped, but functionally, they don't all have the same role. So let's start with taking a look at the pictures. So here, we can see the basal ganglia, and this is the thalamus, and here is the amygdala. And if we zoom more in, here we can see the corpus callosum, this one, okay? Here we can see the putamen and the globus pallidus, and here we can see the caudate nucleus. So again, it's like caudate nucleus, putamen, and globus pallidus. Here is the amygdala, okay? And here you can see the caudate nuclei, the putamen laterally, the globus pallidus medially. Again, it's the putamen is always lateral, globus pallidus is medial, and you can see the amygdala. Let's try to see this picture. This picture is showing us again the caudate nucleus, the putamen laterally, and medially the globus pallidus. Under them is the amygdala, and here we said the sheet of gray matter. Deep to the insula is the claustrum. Here in this picture is showing us what the anterior horn of the lateral ventricle from the ventricular system video. Here we can see what the head of the caudate nucleus, okay. We can see the claustrum here, this gray matter. Here I can see the lentiform nucleus, the putamen, okay. And I can see the tail of the caudate nucleus. So this is the head of the caudate nucleus, and this is the tail of the caudate nucleus. Okay, and this is the insula. So basically, we have the insula, and then the claustrum, and then the external capsule, and then the anterior limb of the internal capsule. So this is from outside to inside, if we're looking at it this way. Okay, and here we have the internal capsule as well on the very inside. Let's move on to get familiarized more with these pictures. So let's talk about the anatomy of the corpus striatum. So the name corpus striatum, it comes by the meaning of body with striations, and it's divided to two major components, the lentiform nucleus and the caudated nucleus. So let's start first with talking about the lentiform nucleus. So the lentiform nucleus is going to be a bioconvex lens-shaped nucleus, and laterally it will be bound by the external capsule and medially by the internal capsule. If you remember in the picture that we've seen here, now we're talking more in depth about it. So if we try to search for it, okay, is it shown here? It's not quite much shown here. Let's see here in this picture again. So here we have what? I have the lentiform nucleus, okay? It's biconvex and lens shaped, biconvex and lens shaped. And laterally we have the external capsule, okay? So let's see, this picture is better. So here, lateral to it, we can see the external capsule, and medial to it, I can see the internal capsule. And this is what the biconvex lens-shaped lentiform nucleus, which is consisting of the putamen and the globus pallidus. The putamen and the globus pallidus. Okay? Okay. So the lentiform nucleus has two parts. Again, as we've seen in the picture, the first one will be the putamen, which will be the darker and the lateral part. So let's see it here in the picture. So the putamen, it will be here, and it will be the darker and the lateral part. The second part of the lentiform nucleus is the globus pallidus, the GP, which is pale, and its medial part. Okay? And the globus pallidus basically is important because it is the efferent or the output part of the basal ganglia. So we call the globus pallidus, let me write it here, we call it the efferent part of the basal ganglia. So if the globus pallidus, for example, degenerate, what will happen when the globus pallidus will degenerate? It will cause something called the athetosis, which is a type of involuntary movement. So this is the first clinical correlation. Athetosis, which is a part of the involuntary movement. And remember that the globus pallidus is divided into what? It's divided into a globus pallidus interna and a globus pallidus externa. So let me see if we can see a more clear image for it. So yeah, so here it can show the internal and the external part, but I'm trying to search for another better image. Okay, so maybe we can see it in another place. It's okay. So again, just put in mind it has an interna and an externa. Okay? Okay. Now, so this is it for the lentiform nucleus. Okay, so here we can see the external capsule on the outside, the internal capsule, which will be medial to it, the lentiform nucleus, forms of putamen and a globus pallidus, external and internal. If you can see these two colors, external and internal. And now let's get moving and let's talk about the second major component of the corpus striatum, which is the caudated nucleus. So the easiest way to recognize it is that it's going to be either C-shaped or comma gray matter, comma shaped gray matter. Okay. Now it has three parts. It has the head, the body, and the tail. So the head, it will basically form for me what? The floor of the anterior horn of the lateral ventricle. So in the anterior horn of the lateral ventricle, the floor of it is formed of the head of the caudated nucleus. And then the body, it will arch up and back. So it will go up and back above the thalamus and the lentiform nucleus. And finally, the tail will curve down into the temporal lobe, ending at the amygdaloid body. So let's try to take a look at it here. So here we have the caudated nucleus, the head, the body, and the tail. Okay, the head and the body and the tail. And what did we say about it? The head form the floor of the anterior horn of the lateral ventricle. The body will go up and back, okay, above the thalamus and the lentiform nucleus, and then the tail, it will go down to the temporal lobe, ending at the amygdaloid body. This is the amygdaloid body here. So it's very important to know that whenever we see C for caudate nucleus, we think of C for chorea. What does this mean? If I have a degeneration of the caudate, it will lead to chorea, which will be seen in the rheumatic fever or the Sydenham chorea, as well as Huntington disease. So I already talked about the globus pallidus, we said athetosis, and now for the caudate, we said chorea. Okay, so keep that in mind. Now moving on, let's stop thinking only about anatomy and let's look at the functional classification. But before that, let's look at some pictures in order to help us with the OSPI. So here, for the horizontal section, we can see again the corpus callosum. Here I can see the head and then the tail of the caudate nucleus. And I can see here the lentiform nucleus formed of the putamen laterally and the globus pallidus medially. And lateral to it, I have the external capsule. Medial to it, I have the internal capsule. This is the insula. And here I can see the thalamus. Okay, and this is the third ventricle in between both thalami. And what else can we see? This is hippocampus, pineal gland. Let's take a look at it in a coronal section on the other hand. So here we can see the lentiform nucleus. Here I can see the colostrum. We said it's the gray matter. And within, the caudate nucleus will be here. This also would be the caudate nucleus to the head and the tail. This is another picture, shows the caudate nucleus. It shows the lentiform nucleus in globus pallidus externa and interna. And this is the putamen outside, obviously. So yeah, and for the picture here, the coronal picture, what can we see in this picture? So we can see here what? The lentiform nucleus, laterally the putamen, and then the globus pallidus. You can see the putamen is darker in color, the globus pallidus is more pale. And here we can see the caudate nucleus. Yeah. Okay, now moving on, let's talk about the functional classification and the blood supply. So there are two important terms here that we need to think of, afferent and efferent. And afferent, it means input, in. And the afferent part of the basic ganglia, it's going to be the striatum, also called the neostriatum. And the striatum consists of what? It consists of the caudate nucleus plus the putamen. Pay attention. We're not talking about the lentiform nucleus. We're talking about a new term here called the neostriatum or the striatum alone. And it means the putamen plus the caudate. We're not mentioning the globus pallidus here. So the putamen plus the caudate. Okay? It mainly receives signal from where? From the cerebral cortex, and then it will send inhibitory or negative signals to the palladium. So again, it will give me inhibitory signals from the cerebral cortex to the palladium. Now, on the other hand, talking about the efferent or the output, the main efferent part here is the palladium, the globus pallidus alone. So here we didn't talk about the GP. Here we're talking about the GP because it's the only efferent one. So the high yield for us to know is that the striatum or neostriatum is input and the output is the GP. And again, let me refresh your memory. If we have degeneration in the globus pallidus, what will happen? Pause the video and try to remember. And if you said athetosis, you're correct. And what is athetosis? Try to remember again. If you don't remember, you can go back to the part where we talked about it. So now let's get going and let's take a look at the blood supply of the basal ganglia. It's very important to know that the main blood supply, it comes from the striate arteries. What do I mean by striate arteries? I mean that they are central or penetrating branches of what? The middle cerebral artery. They're central or penetrating branches of the middle cerebral artery. Now, these arteries, they supply what? They mainly supply the lentiform nucleus and the posterior part of the internal capsule. So they supply mainly the lentiform nucleus, which we said it consists of the putamen and the palladium, and the posterior part of the internal capsule. Very important here to know that these striate arteries, they are actually end arteries. What do I mean by this? I mean, if one of them became occluded, so occlusion happened in one of them, there isn't an alternative blood supply. So I don't have an alternative blood supply. So if I have a damage here, it's going to be severe. Nothing is going to make up for it. There is also an anterior supply that I need to talk about. So we talked about the main supply, which is the striate artery. What about the anterior supply? So the anterior supply, let's talk about the central branch of the anterior cerebral artery. So here we talked about the middle cerebral artery. For the anterior supply, anterior cerebral artery. Okay? So it supplies the small anterior part of what? The head of the caudate, okay? And the anterior internal capsule. Here we said the posterior internal capsule. Here we said anterior internal capsule and the head of the caudate nucleus. Uh, okay, so let's take a look at it here. So we said mainly we have straight arteries, okay, and the small anterior part is by the anterior cerebral artery, the central branches of it, okay? So here we have what? The anterior cerebral artery, and we have the middle cerebral artery divisions. So just familiarize yourself with this and you'll be good to go. Now, let's move on and talk about one of the most important concepts of the basal ganglia, which is what? It's called gating. What do I mean by gating? The basal ganglia, basically, it helps control what? It helps to control the gross and the crude movement, like walking, flexion, the basic movement of human species. They're also involved in another thing, which is the autonomic execution of learned skills. Okay? So, like movement become less awkward with practice whatsoever. So basically, when we first learn a movement, it may feel awkward for us. For example, the first time you're riding a bike, it would feel awkward for you, but with continuous practice, it will become automatic and less awkward. This is what I mean, execution of learned skills. So the basic ganglia, they're also involved in what? They're involved in planning and learning through area 6. But the key concept here, aside from this, is talking about the gating function. So we need to think of the basal ganglia as deciding which movement should be allowed and which movement should be suppressed. So we're talking about a gate, a door. It lets certain movements pass and other movements are not allowed. Okay? So we have two pathways. Let's start first with talking about the direct pathway. So the direct pathway, okay, I'm just simplifying this structure here. So the direct pathway, it helps select and facilitate desired motor activity. Okay, so it's for the desired motor activity, and mostly it involves the globus pallidus interna, the GP interna. Whereas the indirect pathway, from its name, it will suppress the unwanted movement and it will use the GP externa. Okay? So the direct is for the interna, the indirect for the externa. We need to remember that these two, the direct and the indirect, they're happening simultaneously. So the basal ganglia are not simply turning the movement on or off, they're basically helping select the correct movement while suppressing the movement that is unwanted, and this is happening simultaneously. This is what I mean by the gating function. Okay, now let's talk about, or let's apply the gating concept to two important diseases that we need to be familiarized with, the Parkinson disease and the Huntington disease. So first, let's start with the Parkinson disease. The Parkinson is a hypokinetic disease. What do I mean by this? It means hypo, so I have reduced or slowed movement, either reduced or slowed movement. So in Parkinson, what happens in this case, pay attention to this here while I'm explaining it to you. There will be a degeneration of a substance called the substantia nigra. So this will disrupt for me what? This will disrupt for me the inhibitory nigrostriatal dopaminergic system, which will make the basal ganglia hyperactive. As a result of this, what will happen? Desired movement will become, so the desired movement, it will become slowed, while the unwanted movement, okay, they will not be inhibited, not properly inhibited, which will lead to me for the tremors, mainly the pill rolling tremor of the Parkinson. Okay, so what is happening here again? We have a disrupt in the inhibitory nigrostriatal pathway, the dopaminergic pathway, because I have what? I have degeneration of the substantia nigra, leading to hyperactivity, leading to the desired movement becomes slowed and the unwanted movement is not inhibited, leading to tremor. Whereas if we talk about the Huntington disease, on the other hand, it's called the hyperkinetic, means I have excessive unwanted movement. Unwanted movement. So here what will happen? I have degeneration of the neostriatum. What did I say about the neostriatum? Is the caudate nucleus and the putamen, if you remember. So this degeneration of it, it will inhibit for me or disrupt for me the striatonigral pathway. And this will lead to what? Overactivity of the substantia nigra. And the basal ganglia will become hypoactive. Now, because the basal ganglia is failing to inhibit the unwanted movement, they will develop chorea. So we need to remember what? Parkinson is hypokinetic, Huntington is hyperkinetic. So again, I have degeneration of neostriatum, striatonigral pathway disrupted, activation of the substantia nigra, lead to hypoactivity. The unwanted movement is failing to be inhibited, so they will develop for me the chorea. So Parkinson is associated with a tremor and Huntington with chorea. Okay, now let's move on from the basal ganglia and start with talking about the cerebellum. Now, the first major point in order for us to remember here is that a cerebellar lesion will cause uncoordinated and awkward movement, but it will not lead to paralysis. This is very important to put in mind. And when we look at the cerebellum anatomically, it's going to be the largest part of the hindbrain. So it's the largest part of the hindbrain. And it's located where? In the posterior canal fossa underneath the tentorium cerebelli. So in the posterior canal fossa underneath the tentorium cerebelli. It consists of two hemispheres, which are connected in the middle by what? The vermis. So the cerebellum, basically, it connects to the brainstem through three pairs of peduncles. So we have first the superior peduncle, which connects it to the midbrain. Then I have the middle peduncle, which connected to the pons, and they are the thickest, as you can see. And then I have the inferior peduncle, which connects it to the medulla. So it's very important to know the superior with the midbrain, the middle with the pons, which is the thickest, and the inferior with the medulla. And here's how we can see it, okay? So the superior, the middle, and the inferior. This is on the uh, lateral aspect of the brainstem. Okay, now let's take a look at this picture here. So this is the midbrain, the Mickey Mouse shaped. You can refer back to my brainstem video in order to see them. Here I have the anterior cerebellar notch, okay? So anterior cerebellar notch, the vermis cerebral hemisphere, and the posterior cerebellar notch. So I have anterior notch and posterior notch. And this is the top view of the cerebellum, by the way. Now for the other names, here we're going to take a look at them later on. So let's get going. And right now we're going to talk about the three lobes of the cerebellum, and each and every one has a different major function. So let's start first with talking about the anterior lobe, which we also call the um paleocerebellum, the paleocerebellum. The mnemonic here to remember is pale aunties spinning their heads and limbs. Okay, so what do we get from this? It connects the spinal cord to coordinate gross movement of the head and the limbs. This is the anterior lobe. Okay? And then the second one, the middle one, okay, or the posterior lobe, they're called the neocerebellum. So the middle or the posterior lobe, they're called the neocerebellum. And for this one, the mnemonic is going to be the new court volunteer did a fine job mid-trial. Okay? Now, this is the largest part of the cerebellum. It's very important to know that it's the largest part of the cerebellum. And it connects with what? It connects with the cerebral cortex to coordinate the fine voluntary movement of your hands, your eyes, and your larynx. And lastly, I have the flocculonodular lobe, also called the archicerebellum. This is the oldest. So this is the largest, and this is the oldest. Okay, oldest part. It consists of what? One median nodule and two lateral folliculi. So one median nodule and two lateral folliculi. And its main connection is what? It's with the vestibular nuclei. So it's important from its name for the equilibrium and trunk of balance. So again, the anterior lobe, it's the one that we see here, okay? And remember the mnemonics associated to connect to the spinal cord, head, and limbs. Then we have the middle or posterior neocerebellum, the largest part, okay? The middle or the posterior, here, okay? And it's associated with the cerebral cortex for fine voluntary movement of the hand, eye, and larynx. And then for the flocculonodular lobe, the oldest one, we said so we have what? We have one nodule median and two folliculi lateral. So one nodule medially and two folliculi laterally, and it's associated with the vestibular nuclei for the balance. Okay. Let's take a look at this picture here. So as you can see here, we have the cerebellum. This is the cerebellum tonsil, and this is the pons. Here on this picture, we can see the fourth ventricle, the pons, okay, the medulla oblongata. And here, what can I see? The basilar artery. Here, what can I see? The cisterna magna. Here's the cerebellar tonsil. And the cerebellar tonsil is very important to know that it's the lowest part of the cerebellum. So it herniates through the foramen magnum. Another picture here, it shows us here the pontine cistern, which contain the basilar artery. Here I can see the frontal sinus, the sphenoid sinus, the pituitary fossa, the pons, the fourth ventricle, and frontal lobe and the anterior cranial fossa, and the right cerebellar hemisphere. Okay, this is the left cerebellar hemisphere. Coming here to this picture, it shows us the vermis of the cerebellum and the cerebellar hemispheres. Okay. Now, let's take a look at the internal structure of the cerebellum. And if we take like a cross section, we can see what? We can see the um, outer gray matter and inner white matter. So an outer gray matter and inner white matter. Okay, and deep gray nuclei. You can see deep gray nuclei within them. So outer gray matter, inner white matter, and deep gray nuclei. Now, the outer cerebellar cortex, okay, the cerebellar cortex, it has three layers that we need to mention. The first one is going to be the molecular layer, okay? So this layer, the molecular layer. Now, this is the external layer, and it has the stellate cells and the basket cells. So it has the stellate and the basket cells. The second one is the Purkinje layer, okay? So the Purkinje layer, which has the Purkinje cells, and it's the middle layer. And a very important point is that the axons actually of the Purkinje cells, they project to the deep nuclei and they have an inhibitory effect. So very important to know that. Okay, so the axons of the Purkinje cells, they go to the deep nuclei and have inhibitory effect. And finally, we have the granular layer, this layer here, which is the internal layer, which has the granule cells and the Golgi cells. So from the outside to inside, we need to remember it as molecular, Purkinje, and granular. And everyone, you need to know what they have in their cells, like what cells they have. Okay. Now, deep inside the cerebellum, we have the intracerebellar nuclei, and we're gonna also talk about the white matter fibers. So, from lateral to medial, we need to remember the mnemonic of don't eat greasy food from lateral to medial. Okay? So this gives us what? From lateral to medial. So the most lateral, I have the dentate. So it's dentate, emboliform, globose, and fastigial. So laterally, I have the dentate nucleus, which is the largest nucleus. It has a fang-like appearance and is connected to the cerebral cortex. So it's this one, the most lateral one. Next to it is the emboliform nucleus, this one, the emboliform nucleus. So it's oval in shape, as you can see here, and it connects to the spinal cord. And right after that, I have the globus nucleus. So it's going to be the globus nucleus, okay, which is round also, and it also connects to the spinal cord, so similar to the emboliform. And lastly, I have the fastigial nucleus, which is the most medially, the fastigial nucleus. Oh, wait, I made it wrong. So we have the dentate, and then the emboliform, and then globus and fastigial. So it's from here. So lateral to medial. Sorry for the inconvenience again. So last one, which is the most medial one, is the fastigial. Okay. And this is the most medial one, obviously. It connects to the vestibular nuclei and it's involved with the balance. So it's very important to know what each and every one connects to. We also have two important types of white matter fibers, the climbing fibers and the mossy fibers. So the climbing fibers, okay, so here we have the mossy fiber, okay, and here we have the climbing fiber, mossy and the climbing. So the climbing fibers, they come from the olivary nucleus, okay, so they're coming from the olivary nuclei. They enter through the middle or the inferior cerebellar peduncle, okay, and they terminate on the Purkinje cells, and their effect is excitatory. Whereas the mossy fibers, they come from the pontine nuclei and they enter through the middle or the inferior cerebellar peduncle, again the same entry, but they terminate where? They terminate on the granular cells and also their effect is excitatory. So it's very important to know that. Okay, so now let's understand how the cerebellum actually help us to make smooth and precise movement. Now, one important point here is that the cerebral cortex and the cerebellum, they do not communicate directly. Okay, so again, the cerebral cortex and the cerebellum, they don't communicate directly. Instead, they communicate through the pons, specifically through the pontine nuclei. So we need to think of the movement as if like we're talking about a feedback loop. So when the cortex plans a movement, it will send what? It will send for me a planned copy to the cerebellum through the pons. So whenever the cerebral cortex is thinking of a movement, it will send it through the pontine nuclei to the cerebellum. And at the same time, the spinal cord will send the executed copy to the cerebellum through the spinocerebellar tract. So again, the cerebral cortex, it will send to the cerebellum through the pons. And the spinal cord will send to the cerebellum through the spinocerebral tracts. So the cerebellum is receiving two pieces of information, what we planned to do and what we actually did. So what we planned and what we actually did. So the cerebellum compares these two, then it will send the feedback back to the cortex. So once it takes one and two from the cerebral cortex and the spinal cord, it will take this information and give it back where? To the cerebral cortex. But in order to give it back, it will go through a pathway. The pathway will be what? Through the dentate, and then the red nucleus, and then the thalamus, and then to the cortex. So dentate, red nucleus, thalamus, and then the cortex. It also sends back to the spinal cord, but it will send to the spinal cord. So this is first, its response to the first one, cerebral cortex. We said these four steps. Then it will send back to the spinal cord, but it will send back through what? Through the rubrospinal tract and the vestibulospinal tract and the olivospinal tract. So the overall purpose is to correct errors and make the movement smooth, coordinated, and precise. Okay? So again, the cerebral cortex to the cerebellum through the pons. This is exactly what I told you, through the pontocerebellar tract and the corticopontine tract. And then here we have what? Um, So the spinal cord through the spinocerebellar, and then the cerebellum, it will send it back through the dentate, the red nucleus, and the thalamus, back to the motor cortex, and then to the spinal cord through the red nucleus, vestibulo, and olivary. Okay? I hope it makes sense. Okay, so now let's quickly cover the blood supply of the cerebellum. So it's very important to know that I'm going to have here three main arterial supply. The superior cerebellar artery and the inferior cerebellar artery and the posterior inferior artery. So the SCA, the ICA, and the PICA. Okay? Now, the three arteries to remember are these. For the venous drainage, on the other hand, so first let's take a look at them. So again, the SCA, the ICA, and the PICA. Now, for the venous drainage, the superior surface is going to drain towards the straight and the transverse sinus and the great cerebral vein. So this is superior surface, straight, transverse, and the great cerebral vein. Whereas the inferior surface will drain towards the inferior petrosal and the occipital sinuses. So it's mainly just memorization for this slide, honestly. Lastly, very important and most loved in the exams is the clinical correlation. So let's apply everything that we have learned to the clinical presentation of the cerebellar lesions. The main clinical problem we're going to discuss here is motor ataxia. Okay? The most important distinction here is that motor ataxia is not corrected by vision. Why? Because the problem here is with the cerebellum itself. Okay? Cerebellum is damaged. It's not with your eyes that will fix it. So this is different when we're talking about sensory ataxia, like tabes dorsalis, for example, where the patient can correct it by vision. Motor ataxia, on the other hand, they cannot correct it by vision. Now, there are two major types of the cerebellar ataxia. I have the truncal ataxia, also called the archicerebellar syndrome or the midline lesion. And this one involves what? This one involves the vermis and the flocculonodular lobes, the one that we've seen. The vermis in the middle and the flocculonodular lobes, the one nodule and two flocculi on each side. Okay? Now, in children, okay, the common cause is the medulloblastoma, which is a highly malignant tumor, which leads what? To a hydrocephalus that will obstruct for me the fourth ventricle, so medulloblastoma. In adults, is because of alcohol intoxication. Now, the main signs here will be what? It will be unsteady, staggering, wide-based gait, and the patient will sway around while they're standing. So they will not stand straight, they will be swaying around. An important note here is that there is no disturbance of speech, okay? Or the muscle tone, or limb ataxia in this truncal syndrome. So this won't happen here in this case. The second one, which is called the limb ataxia or neocerebellar or hemispheric lesion, this one can be caused by what? By tumor, abscess, ICA and PICA syndrome, the arteries that we've talked about, which will affect the cerebellar hemispheres. Okay? Here, we need to remember a very important rule that the cerebellar effect, they're ipsilateral. Unlike that, whatever we talked about before, there will be here ipsilateral, unlike the cerebral lesions. In cerebellum here, it's ipsilateral. So the finding will happen on the same side of the lesion, okay? So the findings occur on the same side as we've said. Now, the signs of the limb ataxia, it includes several things that we need to talk about here. For example, we have hypotonia and hyperreflexia, producing شو اسمه pendular reflexes, okay? It could be asthenia, which means muscular weakness. It could be kinetic or intention tremor, which appears during movement. And it's different from the resting tremor. So it's kinetic tremor. The resting tremor is for the Parkinson. Okay. It also can be a decompensation of the movement, meaning movement become like broken down movement, or like choppy movement. It's not smooth. Dysmetria, for example, the patient will have difficulty estimating the distance, for example, nose to finger test. Um, Adiadokinesia, which is the patient won't be able to perform the rapid alternating movement. And when walking, the patient will like go towards the affected side. Okay, so they will go towards the affected side when they're walking. And finally, there can be dysarthria, which is basically like not smooth speech, like stacked or chopped speech. So if we need to remember the overall picture, we need to think of the midline cerebellar lesion as in being truncal ataxia, whereas the cerebellar hemisphere lesion is limb ataxia with ipsilateral signs. And the key clinical concept is that cerebellar damage will produce coordination, okay, but not paralysis. And the motor ataxia is not corrected by vision. So that was it for this lecture. I hope it was easy to understand. Thank you very much for